So, good morning, everyone. Am I audible to you? Yeah. So, let's start now. Okay, Ji? So, prokaryotic cell over. Now we are going to talk about eukaryotic cell. Okay. So, where eukaryotic cells are present? Found in members of protista, fungi, plantae, and animalia. Means except monerans, members of every kingdom, according to 5 kingdom, right, have which kind of cell? Eukaryotic cell. Is it clear? So, as I told already, in eukaryotic cell, who is present? Compartmentalization present. Compartmentalization present. Compartmentalization I already explained to you. Due to membrane-bounded organelle, presence of membrane-bounded organelle, cytoplasm divided into many small compartments. So membrane-bounded organelles are present. Membrane-bounded organelles are present. Getting the things? After that, what you will find? Cytoskeletal structure. Cytoskeleton are present. Microtubule, microfilament, intermediate filament, all these things are present. Is it clear? Well organized nucleus is there. Well defined nucleus. This is present. After that, what you will find? Chromosome, DNA organized in which one? Chromosome. So true chromosome. How DNA organized in chromosome? Have a look in minute. This is a DNA. This is histone protein. Okay, ji? This is histone protein. And DNA wrapped around which one? Histone protein. Right. Then it is DNA. After wrapping, it is called chromatin. Chromatin, when it go for condensation. Condensation. Who is going to form? Chromosome is going to form. It is what basically chromosome is forming. Chromosome is forming. So this is about the eukaryotic cell. There are 2 types of eukaryotic cell I told already, plant and animal cell. And about the difference between plant and animal cell we already discussed. Is it clear? Is all right? Right now, we already discussed about plant cell and animal cell in the last class only. Okay, ji? Done? Over? So who is over? Eukaryotic cell, a basic introduction over. Now we are going to deal about components of the cell, and we are starting with cell wall. Cell wall. So if you talk about cell wall, it is present in, we are going to talk about eukaryotic cell. So I am not including here what? Monera. Already cell wall of monera or prokaryotic cell I told in earlier classes. Na? Is it? So here I am saying in the reference of eukaryotic cell. So it is present in which one? Some protist. It present in fungi. It's present in plantae. Getting the things. So cell wall is present in how many eukaryotic kingdom? If someone going to ask a question, it is present in the member of 3 eukaryotic kingdom. These cell wall not found in Animalia. Not found in Animalia. Clear? And I already told about in the last class regarding the cell wall of what? Monerans. It is non-cellulosic, that is made up of which one? Peptidoglycan. Able to understand or not? So if we talk about in protistans, if cell wall is present, it is cellulosic. Cell wall is made up of which one? Cellulose. In fungi, cell wall is made up of which one? Chitin. It is non-cellulosic cell wall. And in plantae, cell wall is what? Cellulosic. And if I wrote here Monera as well, Monera as well. So in Monera also, cell wall is non-cellulosic. So examiner also asks question, according to 5 kingdom system of Whitaker, cellulosic cell wall present in reported in member of how many kingdom? Cellulosic cell wall are reported in member of how many kingdom? 1 in protistans, 1 in plantae. Getting the things? Cellulosic cell wall is not present in fungi and cellulosic cell wall is not present in which one? Monerans. Getting the idea? So how many kingdom? 2 kingdom. Be careful here. Are you getting the idea? Understanding the idea? Is it clear? So if we talk about more about the cell wall, so we can write like it is outermost protective covering of cell. It is outermost protective covering of the cell. It is a non-living boundary. It is a non-living boundary. Cell wall is a non-living structure. You are getting the idea, right? So what function is cell wall performing? What function is cell wall performing? Function number 1, protection. Function number 2, providing shape of cell, shape to cell. Getting the things or not? Then you will talk about another function it is providing is which one? Another function it is protecting from infection. Getting the things or not? And transport of water and a small molecule. You just keep in your mind, if someone asks a question, cell wall is a permeable structure. Permeable structure allow both solute and solvent to pass through it. So it help in which one? Transport. Getting the things or not? So it is a permeable structure until unless some another material not deposited on the cell wall. If suberization occur, lignification occur, in that case cell wall become impermeable. But that is a special condition. If anyone going to ask you a generalized question, कि cell wall is permeable, semi-permeable, impermeable, selectively permeable, then what the answer will you give? Cell wall is a permeable structure. Getting the idea? Clear in the mind? Okay, ji, is done, over. Now we are going to move ahead. Have a look here. So, if we talk about, because NCERT told about algal cell wall. Algal cell wall. What are the compositions of the algal cell wall? Composition of the algal cell wall is cellulose, galactans, mannans, and mineral like calcium carbonate. These are present in the cell wall of which one? Algae. Cellulose, galactans, mannans, and mineral like calcium carbonate. So students, don't confuse yourself. Hemicellulose is not written in algal cell wall composition. It doesn't mean hemicellulose is not present. Many times teacher also do mistake during framing the question. Right? They find in algal cell walls hemicellulose not written. So they wrote hemicellulose is not present in whose cell wall, and they give the answer algal cell wall. It is a wrong framing. If anything not written is doesn't means it is absent. Are you getting the idea? So what written in NCERT? Cellulose, galactans, mannans, and minerals like calcium carbonate. Hemicellulose is not mentioned. It doesn't mean it is not present. Getting the idea? Clear? It's all right? You got the idea what I want to convey? Okay? Right. Now I am just telling in plant cell wall. Plant cell wall means higher plant cell wall. Algae is also a plant, higher plant cell wall. What NCERT wrote the composition? It is made up of cellulose. It is made up of cellulose, hemicellulose, pectin, and protein. Pectin and protein. Able to understand? These are written in which one? The compositions of the which one? Higher plant cell wall and composition of the algal cell wall. Able to understand? Right? Now we are just going to talk somehow more about the cell wall. I am just telling you how the cell wall is forming. Is it clear? Understand? The outer, so if we talk about the outermost boundary, the outermost boundary or layer, better to say, outermost layer of cell wall is middle lamella. Middle lamella. Why these middle lamella is also called cell cement. It is cementing material between two cells. Means for two cells there is a common middle lamella, and these middle lamella composed of calcium and magnesium pictate. Getting the things? Who is present in the middle lamella? Calcium and magnesium pictate is there. So below the middle lamella, or better to write inside of middle lamella, primary cell wall is present. Primary cell wall is present. Able to understand? If we talk about primary cell wall, is thin and elastic. Thin and elastic. And you also keep in your mind, in meristematic cell, only primary cell wall is present. Only primary cell wall is present. In the case of which cell? Meristematic cell. Getting the idea? Only primary cell wall is present. Now what happened? In mature cell, formation of secondary cell wall occur inside of primary cell wall. Inner side of primary cell wall. Inside matlab inner side of primary cell wall. And these secondary cell wall is of 3 layers, S1, S2 and S3. Getting the things? And this secondary cell wall is thick and rigid. So the rigidity you find in the cell wall is due to which one? Secondary cell wall. In tracheids of gymnosperm, tertiary cell wall also present. Where tertiary cell wall also present? In tracheids of gymnosperm. Able to understand or not? Is it clear? Now let me tell you about the structure of the cell wall. Give your attention. So I am drawing the structure of the cell wall. Assume this is a plant cell. I am telling a very important thing. So connect here. You will understand a lot of things here where you are just missing. Have a look. This is a plant cell. So when plant cell undergoes in mitosis, divide, what happened? When plant cell, what happened? Divide. First karyokinesis occur. Means one nucleus divided into how many nucleus? 2 nucleus. One nucleus divided into 2 nucleus. Getting the idea? Understand up to here? Now we are going to deal about how the cytokinesis occur, how the formation of cell wall occur. Getting the idea? Between the 2 cells. Earlier it was 1 cell. Now it divided and nucleus division occur. Nucleus move on 2 sides. Able to understand or not? So how cytokinesis occur? Is it clear? Now I am telling you, in plant cell actually what happened? Any cell, endoplasmic reticulum present in all over the cytoplasm. So here you look. Here it is. Endoplasmic reticulums was present in plant cell like this. Who was present? ER was present like this. I am telling you the mechanism. Connect with me. So this is the endoplasmic reticulum that was spreaded in all over the cell. Nucleus came this side, another nucleus came this side. Now how the new cell wall form between these 2 cells? Nucleus will separate, na? Getting the thing. So look about the mechanism part. So what's happened? First of all, formation of fragmoplast occur. Fragmoplast assemble like this. Who assemble? Fragmoplast formation occur. These fragmoplast assembling like this. They are moving from center to periphery. These are the fragmoplast. Are you getting the things? What I am writing the name? Fragmoplast. Right. What are present in this fragmoplast? Who is contributing in the formation of this fragmoplast? So I am telling you the compositions of this fragmoplast. This fragmoplast is made up of microtubule. It is made up of microfilament. It is made up of vesicle of Golgi complex. Golgi complex. And also it is made up of which one? ER. Component of ER. You got the idea? So how many things together forming these all assembled together and forming the which one? Fragmoplast. Is it? And these phragmoplasts are the scaffold of the frame of the which one? Same plate. They are forming the frame. Able to understand or not? So later on, these phragmoplasts converted into which one? Cell plate. Later on, these phragmoplasts converted into which one? Cell plate. Are you getting the idea, understanding my point, what I want to say? Where this phragmoplast is not there? From where endoplasmic reticulum part move. Lumens of the endoplasmic reticulum move. There who is not there? Fragmoplast is not there. So where fragmoplast not deposited, in that area who will form, dear children? Pits. Pit formation. Able to understand? What we are finding? Wherever pit is there, cavity is there, na? Cell wall is discontinuous. Cell wall is not there. So pit is forming. So even why pit is forming? There is a tube. You just think like there is a pipe going. And you are just making a wall. So you can't put the bricks in the wall, na? In the pipe. Na? You can add bricks this side of pipe and this side of the pipe. You can't add the bricks in the pipe. Getting my point, what I want to convey? Is it clear? Understanding the idea? Yeah. So what I am saying, when cytokinesis occur, a plant cell divide, first of all, what happened? Fragmoplast, now it is A cell, it is B cell. Fragmoplast assemble between the two cells. And these fragmoplast is made up of microtubules, microfilament, vesicle of Golgi complex, and ER. All are together contributing in formation of this fragmoplast. Later on, these fragmoplasts converted into cell plate. Later on, this cell plate converted into middle lamella. So entire journey, did you understand here or not? Entire journey, did you understand? Is it clear how the middle lamella is forming? First fragmoplast formed by these four components, then it converted into cell plate. Now it converted into which one? Middle lamella. Middle lamella is formed from cell plate only. And now this is cell A, this is cell B. I am telling these middle lamella now is a common wall between these two or not? Like cement are present between two bricks. You will say cement belongs to both the bricks, na? Same way. These middle lamella present between A cell and B cell. Is it clear in the mind? You are getting my point, what I want to convey, dear children. Is it all right? Now what happened? Both side, inside A and inside B, formation of who occur? Primary cell wall. This side primary cell wall, this side primary cell wall. Primary cell wall. Are you getting the idea? Primary cell wall is forming. Then primary cell wall, as long as cell remain meristematic, only primary cell wall is there. After the primary cell wall, three layers of secondary cell wall will form. One layer, two layer, three layer. This side also, one layer, two layer, three layer. Here also one layer, two layer, three layers. This side also one layer, two layer, three layers. Secondary cell wall. So secondary cell wall is three layered structure. Who is there? Thick structure. And that is the reason your textbook also wrote like this. Whenever the formation of secondary cell wall occur, primary cell wall get diminished. Because when a thin layer was there, as a thick layer came, who become more visible? Thick layer become more visible, na? Understanding the idea? Getting the idea? So who get diminished? Primary cell wall get diminished. Is it? Are you getting the idea? And in some track, so it is primary cell wall. It is P. It is S1. It is S2. It is S3. It is P. It is S1. It is S2. It is S3. These all three are secondary cell wall. So what are the primary cell wall present for both cells. But who is common for both cells? Middle lamella is common for both cell, and that is the reason we call middle lamella as a which one? Cell cement, and it is made up of calcium and magnesium pictate. And these endoplasmic reticulum, who is common passing from one cell to another cell, na? Right? This lumen is called desmotubule. Desmotubules. And wherever cell wall material not deposited, that area is called what? Pits. So why pits are forming? Are you getting the idea? Because in the way of the boundary formation, a pipeline is coming. So how boundary forms? Inside the pipeline. And due to that reason, who is going to form? Pits are going to form. Is it clear in the mind? You understand about this cell wall? Is it clear? It's all right? Can we move ahead? Yes. Now, I am just telling you about one more thing where students have confusion. They simply mug up the things. And that thing is what? Plasmodesmata. That thing is what? Plasmodesmata. Is it clear? Can we move? Okay, ji. Have a look. So you just look here. You just think like this is the cell wall. And here who is there? Pit. Who is present? Pit. A side of the cell, B side of the cell. And these are the cell wall between two cells. And where I not made the cell wall, continuity we not made is what? Basically pit. Let me draw another thing. Now this is cell membrane. Who is this? Cell membrane. Right. This blue color one. This is cell wall. You just got it. Just have a connectivity with me. And it is cell membrane. It is cell membrane. Okay, ji. It is done. It is over. Now, here, it is desmotubules. Who is this? Desmotubules. This is desmotubule. So these desmotubules is passing through the pits. These desmotubule is what basically? It is a modified, a smooth endoplasmic reticulum. If someone asking more precise question. So it is what modified smooth endoplasmic reticulum. Is it clear? So it is desmotubules. Done. Now you are finding there is some gap there, and this is cell, one cell, and this is what? Another cell. You just try to understand. This is cell number 1, this is cell number 2, and these are cell wall in between. Is it clear? Now what I am telling, there is a gap there, na? There is a gap in between desmotubule and cell membrane. Gap is there or not? Tell me, some gap is there or not? I am just magnifying it. You will find. Here gap is there or not? In between cell wall and cell membrane, in between desmotubule and cell membrane, there is a gap. So this gap is called cytoplasmic sleeve. Cytoplasmic sleeve. This is called cytoplasmic sleeve. Now, here it is the cytoplasm of the one cell. Cytosol, it is the cytoplasm of the one cell, and it is the cytoplasm of the another cell. Everywhere you just think like this. So these cytoplasm are in continuity in both the cell. You are getting the idea or not? These cytoplasm are in continuity. So from these cytoplasmic sleeve, water, sugar, amino acid means these small substances, means micromolecules, exchange easily between the two cells. Exchange easily between two cells. Because their cytoplasm is in continuity, na? Getting the things? So in between two cells, cytoplasm are in continuity. Both the cytoplasm, there is a small area, na, hole, through which cytoplasm easily moving from this cell to this cell. Who is present in cytoplasm? Easily moving from this cell to this cell. And they are establishing the, which one? Interaction between the two cells. They are establishing the interaction between the two cells. Two cells are exchanging the material through this cytoplasmic sleeves. Are you getting the idea or not? So plasmodesmata is made up of three things. How many components present in the plasmodesmata? Three components: plasma membrane, desmotubule, and cytoplasmic sleeves. How many components are there? Plasma membrane, desmotubule, and cytoplasmic sleeve. All together it is called plasmodesmata. So many plasmodesmata, hundred or thousands of plasmodesmata present between the two cells in the plant cell, and they are making the easy access or availability of the material from one cell to another cell. Are you getting the idea? Understanding the idea? So you got a clarity about the plasmodesmata or not? Tell me, dear children. Got it? See, na, this is cytoplasm. So small substances like water, sugar, amino acid easily diffuse from one cell to another cell. Clear? Over, right? But bigger things can't cross. Bigger things can't cross. Big protein can't cross, na? Because that cytoplasmic sleeve is not that much big. Is it clear? It is all right? Is it all right? So who is over? And it is also gated. I am also telling you something. Here in the cell wall, callose deposition going to occur. And these callose is regulating the plasmodesmatal closure and opening. So when there is no requirement of exchange, when cell don't want to take anything from another side and want to give the other side, what happen? These callose is going to block the which one? Cytoplasmic sleeve. And when cytoplasmic sleeve block, transport is get stopped. So it's not like always exchange occur. Regulated exchange occur. So these callose plugs, these callose is helping in which one? Regulation through the regulation of transport through which one? Plasmodesmata. Clear? They are responsible for regulation of transport through plasmodesmata. So in plasmodesmata, how many components I told? In plasmodesmata, three things are there. Plasma membrane, first component, desmotubule, tubules, and third one is what? Third one is what? Cytoplasmic sleeve. And it is passing through what? Pits. Getting the idea? So plasmodesmata is responsible for exchange of material. Exchange of material between two cells. Between two cells. Got the idea? Right? Clear? Done? So cell wall over. You got the idea? Cell wall over finally. Now we are coming on plasma membrane. So come on which one? Plasma membrane. Plasma membrane or cell membrane are same thing. Both are same thing. Sometimes it is also written at plasma lemma. In protozoans, we use the term na plasma lemma. So it is the modified simply name of the which one? Synonyms terms used for which one? Plasma membrane. Now we are moving ahead. So plasma membrane, it is a thin and elastic membrane. It is a thin and elastic membrane. Okay, ji? So this plasma membrane is thin and elastic membrane. It is universally present in every cell. Universally present in every cell. You cannot tell me a single example, a cell who is living and that don't have plasma membrane. Getting the idea? You can't say it. Without plasma membrane, existence of life of the cell is not possible. So if any living cell is there, by default, who will be there? Plasma membrane is there. Able to understand? Clear? So in plant cell, it is present below the cell wall. Below means inside. Below means inside. Got the idea? I am writing below in the reference of inside. Below the cell wall means inside of the cell wall. And if you talk about more about plasma membrane, so it is selectively permeable membrane. It is selectively permeable membrane. Now question arises, what is selectively permeable? Selectively permeable membrane allow solvent and selected solute to pass through it. Pass through it. Able to understand? So it is a selectively permeable membrane. Is it clear in the mind? It allows solvent, means water can pass from plasma membrane. It not stop the water to pass, but it not allow all solute to pass. It accepts some, allows some, rejects some. Getting the things or not? So it is given the gated entry to whom? Solute. You are in, you are out. They are selective in nature, but it allow every solvent. Getting the things or not? You can say about water, if you talk about a solution, so it allow which one? Selected solute as well as solvent. So it is selectively permeable membrane. Now we are going to talk about composition. Compositions of the plasma membrane. So if we talk about chemical composition of the plasma membrane, it is made up of lipid. Also who is there? Protein. Also who is there? Carbohydrate. All three are present in the plasma membrane. Lipid, protein and carbohydrate. All three are there. Getting the idea? So if we talk about in human RBC, plasma membrane, protein is how much percent? 52%. And lipid is how much percent? 40%. So the percentage of protein and lipid is variable in different plasma membrane. Percentage of protein and lipid is variable. It is not fixed data. This data is applicable for mammalian RBC, human RBC. Protein and lipid content is variable. Variable in the plasma membrane. Getting the things? Is it clear in the mind? Right, Ji? If we talk about carbohydrate, it is 2 to 3%. Very good. The value of the carbohydrate is how much? Amount, compositional percentage of the carbohydrate is how much? 2 to 3%. Now we are going to come on lipid part of the plasma membrane. Which part of the plasma membrane? Lipid part. lipid part of the plasma membrane. If we talk about lipid part of the plasma membrane, following lipids are present. Phospholipid, phospholipid. Able to understand? Who is present? Phospholipid. Another who is present? Cholesterol. Cholesterol. Who is also present? Sphingolipid. Sphingolipid is also present. Able to understand? So phospholipid, cholesterol and sphingolipid are also present in the lipid component. So in all the lipid, major component, major lipid is which one? Major lipid is which one? Phospholipid. Able to understand? Getting the idea? Getting the idea? Major lipid is which one? Phospholipid. Is it clear in the mind? Okay, ji. Right? These phospholipid is also, you are saying what? Phosphoglycerides. Phospholipid, we are also saying what? Phosphoglyceride. Is it clear in the mind? Okay, ji. Now I am talking about phospholipid. I am talking about phospholipid. So if you talk about phospholipid, it is present as bilayer. Getting the things? It is present as a bilayer. So this is a phospholipid. Okay, ji, this is a phospholipid. Here it is fatty acid 1 chain, it is fatty acid 2 chain. And in head who is there? Glycerol plus phosphoric acid. Phosphoric acid is present. So this is the phospholipid. Head part is made up of glycerol plus phosphoric acid and tail is made up of fatty acid. So here 2 fatty acid chain are there attached with a glycerol plus phosphoric acid. With glycerol, 2 fatty acid is joined. So what I am telling, in this phospholipid, with glycerol, 2 fatty acid, and 1, which one? Phosphoric acid joined. Phosphoric acid joined in the case of this phospholipid. Along with glycerol, how many? 2 fatty acid and 1 phosphoric acid joined. You will got this idea in much better way when I will teach you which one? Biomolecule chapter. There I will also draw the structure. See, I told you na, with glycerol, 2 fatty acid and 1 phosphoric acid joined. Getting the idea? Is it clear in the mind? Is it all right? It is clear, right? So have a look here. So head of the phospholipid bilayer is hydrophilic. And the tail part, this fatty acid chain is called tail, and this tail is hydrophobic. Hydrophobic. So these hydrophobic fatty acid chain may be saturated and unsaturated. NCERT only wrote which one? Saturated fatty acid. But it also which saturated fatty acid is there? Unsaturated fatty acid. So fatty acid chain either saturated or unsaturated. Saturated means double bond not present, and unsaturated double bond present in the chain of the fatty acid. You know na, fatty acid is like this, COOH. So if a double bond is there in the long hydrocarbon, it is called unsaturated, and if a double bond not there in the long hydrocarbon chain, it is called which one? Saturated. Saturated, and here it is what? Unsaturated. Getting the idea? Understanding the idea? So what I told you about the phospholipid, phospholipid have what basically? Head and tail. In head who is there? Glycerol. and along with that phosphoric acid attached. And to tell it is a cartoon model, you are finding is the chain of the fatty acid. The head is hydrophilic and the tail is hydrophobic. And that is the reason the fatty acid present in bilayer. The fatty acid present in bilayer, it is present like this. It is present like this. It is present like this. So these fatty acid present in bilayer. Suppose that this side water is there. This side water is there. So what they are doing? What these are doing? They are pushing their head toward the water, in the side of water. And both the tails are together, that is away from the water, because tail is hydrophobic, they protect it from the water, and that is the reason it is present away from the water, and both tail join together. Getting the things? Both head facing outside, both tail facing inside. Are you getting the idea? So hydrophobic tail are together, hydrophobic head is toward outside. Here it is, suppose that outside, here it is inside, and it is the membrane inside of the cell. Who is there? Cytoplasm. Here also fluid is there. Outside of the cell also who is there? Extracellular fluid. Extracellular fluid. Are you getting the idea? So these are the tail. They are protecting their tail. They are protecting their tail from fluid component which is present outside the cell and fluid component present inside the cell. You are getting the idea? So why phospholipid is present in bilayer? You understand the things or not? Tell me, dear children. You understand the things or not? You understand the idea or not? Yes. Now I am telling something more. Have a look. Though these phospholipid bilayer, these phospholipid bilayer is forming the structural framework of plasma membrane. A structural framework of plasma membrane. Who is forming the structural framework of plasma membrane? Phospholipid forming a structural framework of plasma membrane. And one very important thing I am writing. The quasi-fluid nature of plasma membrane is due to phospholipid bilayer. Phospholipid bilayer. Quasi-fluid nature. And that is the reason, dear children, I was saying you one line. The line was what? Saturated and unsaturated fatty acid both are present. So unsaturated fatty acid are what basically? Liquid. Getting the things or not? Liquid somehow. They're not solid. Able to understand? Saturated or solid. So the fluid nature of the lipid bilayer, you will talk about phospholipid bilayer, it is like fluid, it is like honey, it is not solid layer. So that is the reason you cannot cut the plasma membrane. You cannot cut the plasma membrane. You cut the water, you take a knife, cut the water. What happened? Once again they will join. So elasticity, able to understand? These all are due to which one? Quasi-fluid nature. So quasi-fluid nature of plasma membrane, liquid nature of plasma membrane, quasi-fluid nature is due to phospholipid bilayer, and quasi-fluid nature of plasma membrane is facilitating first thing I am writing what? Lateral movement. Lateral movement of protein inside of lipid bilayer. What is this lateral movement? I will tell you, don't worry. Any solid can't allow the lateral movement. Are you agree? Any solid can't allow the lateral movement. Lateral movement allowed by the fluid. Try to understand with the help of an example. Let me tell you. Try to understand it with the help of example about the lateral movement. Give your attention. I am taking a wooden block and taking a nail and beating with the hammer. In wooden block, nail pierced. Now I want to drag the nail here and here. Can I? Can I drag it? Tell me yes or no. Not possible. Now I am taking a bucket of water, putting a stick in the water. Can I drag it? Yes. This is called lateral movement. Did you understand? Is it clear? So lateral movement is possible in fluid. Lateral movement is not possible in which one? Solid. And lateral movement of protein. When facilitated diffusion occur, protein going for rotation. So when anything is going to rotate in the liquid, anything you can rotate in the liquid, but anything can't rotate inside the solid. Able to understand? So these are quasi-fluid nature of the lipid bilayer allowing these things. Clear? Now I am telling. It providing which one? Elasticity. So elasticity have a role in which one? Cell growth. Have a role in which one? Cell growth. Elastic is there, na? They will expand. Able to understand? It is fluid nature is behind the elasticity, much elasticity of the plasma membrane. Fourth, I am writing formation of intercellular junction. Intercellular junction. In tissue, your sir, example, your sir will taught, zoology teacher will taught tight junction, gap junction. Gap junction. Getting the things or not? These all are possible due to which one? Due to which one? Fluid nature of the plasma membrane. Secretion. And you can say exocytosis and endocytosis. Exocytosis and endocytosis. Endocytosis is of two types. Phagocytosis, pinocytosis. Suppose that this is a plasma membrane of amoeba and here is a solid food. So they are just going to cover the food and taking the food inside. This is called which one? Phagocytosis. This phagocytosis is also called cell eating. And suppose that this is a plasma membrane of amoeba and here is a liquid fluid. They invaginate the plasma membrane and food will enter in. It is called pinocytosis. Both are a kind of endocytosis. It is called cell drinking. So these all activity, secretion is a result of which one? Exocytosis. Something is coming out of the cell. It is what? Secretion, na? It is exocytosis. Is it clear in the mind? So exocytosis, endocytosis, secretion, formation of intercellular junction, cell growth, elasticity, lateral movement, all are possible due to which nature of the, of course, cell division also. Also you can write cell division. Of course, cell division also. All these are possible due to which one? Fluid nature of the plasma membrane. During cell division in animal cell, what happened? Plasma membrane invagination occur. So this invagination is possible due to which one? Is fluid nature. Invagination not occur in cell wall, na? Cell wall not invaginate, na? Plate formation occur, na? Getting the idea? Understanding the idea? Is it clear in the mind? Right? So these are the role of which one? Phospholipid bilayer. So now these phospholipid bilayer have one demerits. These phospholipid bilayer in plasma membrane have one demerits. And what one demerit is there? One demerit is there. It allow only which one? Hydrophobic substances. Hydrophobic, or you can say non-polar substances to cross. Not allow polar substances. Not allow which substances? Polar substances. Polar substances. You are getting the idea or not? So this is the demerit. Through phospholipid bilayer part, only hydrophobic and non-polar substances will pass. Polar substances, hydrophilic substances cannot pass through which one? These phospholipid bilayer. Are you getting the idea? Understanding the idea? So is it a demerit? Na? It is allowing a only restricted thing to pass through it. I will tell you later how these polar substances cross. Don't worry about it. Is it clear in the mind? Right? Now, who is over? Phospholipid is over. Who is over? Phospholipid bilayer is over. It is one of the structural frame of the plasma membrane. Are you getting the idea? Understanding the idea? Now I am telling about the another lipid, cholesterol. If we talk about cholesterol, it is more solid than phospholipid, and it provide a stability to plasma membrane. Who provide stability to plasma membrane? Cholesterol provide the stability to plasma membrane. You got about the cholesterol? Got the idea what the job is doing? Now I am telling an interesting thing. Plasma membrane have which one? Phospholipid. Try to understand. I am just telling you, it's a very interesting thing. Plasma, not movement means stability up to, you will understand. One student writing means no movement, sir. Let me tell you. Let me clarify in the better way. If cholesterol is not there, what happened? Plasma membrane in summer season become much more fluider. And plasma membrane become much more fluider, na? Because in summer temperature is more. Who is there? Fatty acid is there. Fatty acid will dissolve and fluidity will increase. So it acts as a buffer for fluidity, not allow to become it more fluid in nature. Able to understand? Getting the idea? Not allowed to become more fluid in nature. Otherwise, the shape get distorted. Na? Are you getting the idea? Understanding the idea what I want to convey? So it is maintaining as a buffer for the fluidity. Otherwise, in summer season, you see na fatty acid become liquid, more liquid in winter and solidify, more liquid in summer and solidified in winter. Who is helping? Cholesterol is helping to get it more fluider. Are you getting the idea? Clear? Now I am telling you about protein component. Sphingolipid I am not going to tell you because it is not your cup of tea at this level. So simply I told a sphingolipid also present in which one? Plasma membrane, but I am not going to tell how much to go detail. Look here now. But plasma membrane is important. That is the reason I am teaching it. Now you just talk about protein. So protein. So in plasma membrane, two type of proteins are present. Two type of proteins are present. One is extrinsic protein. Extrinsic protein. And it is based on ease of extraction. What is the criteria? Based on ease of extraction. How easily it can remove is ease of extraction. Extrinsic protein, or it is also called peripheral protein. Peripheral protein. Getting the idea? And another is intrinsic protein. Intrinsic protein. And these intrinsic protein also called integral protein. So two type of proteins are there. So extrinsic protein or peripheral protein easily removed, removed easily. Able to understand? These are partially buried in the plasma membrane. Partially buried in plasma membrane. And if you talk about intrinsic protein, integral protein is of two types. One is totally buried and it is called transmembrane protein. Transmembrane protein. Getting the things. And another is partially deeper buried. Partially, or you can say deep but partially buried. Deep but partially buried. Got the idea? So let me tell you once again on this. There are two types of protein based on the ease of extraction present in the plasma membrane, extrinsic or peripheral, intrinsic or integral. And if you talk about intrinsic, I am writing, changing the word so you will not confuse. Intrinsic protein or peripheral protein are superficially buried. Superficial word I am using. Very superficial, just above. It is extrinsic protein. It is easily removed. The protein who are intrinsic, not easily removed from plasma membrane. Not easily removed from the plasma membrane. So it is of two possible type. Intrinsic protein is deep but partially buried, not totally buried. And another is what? Totally buried or transmembrane protein. These transmembrane protein are of variety of type. These transmembrane protein are carrier protein. Example of transmembrane protein, carrier protein. Another example of transmembrane protein is channel protein. Channel protein. Getting the things or not? Or tunnel protein. Tunnel protein are the example of this. An example of another transmembrane protein is pump protein. Pump protein. Which protein? Pump protein. Getting the things or not? So these all are the example of which one? Transmembrane protein. Carrier protein, channel protein, and pump protein. These two protein participate in facilitated diffusion. Diffusion. And these pump protein participate in active transport. Active transport. Got the idea? Now everything you look through a diagram. I told about protein. Everything you look through a diagram. So look the classification of protein once again. What I told, protein extrinsic, intrinsic. Extrinsic is also called peripheral. Superficially buried, I will show through a diagram. It removed easily. Intrinsic protein. So these extrinsic protein, what function they are doing? They have enzymatic role. Enzymatic function they perform. Able to understand or not? Now intrinsic protein is of two types, deep but partially buried or totally buried. This totally buried means present from one side to another side of the membrane. It is called which one? Totally buried or transmembrane. It is example of this totally buried are carrier protein, channel protein, pump protein. This carrier protein and channel protein involved in facilitated diffusion. Pump protein involved in which one? Active transport. Getting the idea? Is it clear in the mind? Is it? It is all right. So can I say you the diagram after this protein? Okay, ji. Now I am drawing the diagram. You will understand it better. I am drawing the diagram. Now we are going to talk about a structure of plasma membrane. A structure of plasma membrane. A structure of plasma membrane. So if we talk about the structure of the plasma membrane, what you will find here, best accepted model, many different model is given. So best model for structure of plasma membrane given by Singer and Nicholson, and in 1972. And what was the name of the model? It is fluid mosaic model. Fluid mosaic model. It is given by Singer and Nicholson in 1972. Fluid mosaic model. Right? Is it clear? It is all right. Now I am telling you what he told in fluid mosaic model. Fluid component of the plasma membrane made by phospholipid. And in phospholipid, protein present as mosaic. Protein forming which one? Arranged discontinuously. And fluid component is continuous, phospholipid forming a continuous layer. So in plasma membrane, phospholipid forming a continuous layer. In that continuous layer, protein is embedded here and there like a mosaic. And that is which one? Fluid mosaic model. I am telling you fluid mosaic example. Fluid mosaic example is iceberg in ocean, where oceanic water is fluid, lipid, and iceberg here and there is what? Protein. Are you getting the idea, the meaning of fluid mosaic? Now I am telling another example of fluid mosaic. Gulab jamun in sugar syrup. Sugar syrup, you have seen na? It is fluid, lipid, equivalent to lipid, and gulab jamun embedded here and there, it is equivalent to protein. And this is called which one? Fluid mosaic model. Is it clear in the mind? Now I am telling you the structure of the plasma membrane. Okay, ji, I am drawing the structure of the plasma membrane. Look here. It is phospholipid bilayer. It is phospholipid bilayer. Now here I am just drawing a protein. Then also I am drawing a phospholipid bilayer. You tell me this protein is a which type of protein? It is a extrinsic protein, peripheral protein. Agreed or not? It is an extrinsic protein or peripheral protein. Now I am drawing another protein here. This is from one side of the membrane to another side of the membrane, and this is which one? Transmembrane protein. It is an example of which one? Intrinsic protein. So can I say it? Transmembrane protein. Transmembrane protein. Getting the idea or not? It is an example of which one? Type of which one? Intrinsic protein. It is a type of which one? Intrinsic protein. Later on, further I am making phospholipid bilayer. So see, here and there protein is embedded. This protein is also which one? Extrinsic protein. This protein is also extrinsic protein. Are you agree or not? This is also extrinsic protein. Now I am drawing another protein. Now I am drawing another protein. Look here. Here it is protein. It is intrinsic protein, but superficially varied, not partially varied, not totally varied. Are you getting the idea? It is intrinsic, deeply bent, but not from one side of the membrane to another side of the membrane. So partially varied intrinsic, totally varied intrinsic, and it is which kind of protein? Extrinsic protein. Understanding the idea or not? So these transmembrane protein, I told example for transmembrane protein is what? Carrier protein, channel protein, and tunnel protein. So it is intrinsic protein. Can I say or not? It is also which one? Extrinsic. Not say who are outside. It is outside of cell and it is inside of cell. Inside of cell, and this is plasma membrane. The thickness is 75 angstrom. The thickness is 75 angstrom. Able to understand or not? Is it clear in the mind? So this is which one? Intrinsic protein. Intrinsic protein. Now I am telling you, in the outer layer of the plasma membrane, in the outer layer of the plasma membrane, with protein oligosaccharide attached. It is called glycoprotein. It is called glycoprotein. Getting the things? With lipid, some another oligosaccharide attached, it is called glycolipid. Glycolipid. So these glycoprotein, glycolipid present only toward outer side of the membrane. And toward the inner side of the membrane, another protein is present. It is called aspartame. It is called aspartame. It acts as cytoskeleton. It performs a skeletal role, support a skeletal role. And if you talk about these glycoprotein and glycolipid present in the outer surface, both glycoprotein and glycolipid have a role in act as receptor. Act as receptor. These are also have a role in recognition of self and non-self. And non-self. Who is self, who is non-self? Means what happened? When grafting occur in your body, outside cell get recognized, na? So these outside cell get recognized by this glycoprotein. When fertilization occur, a sperm or egg, same species fertilized, na? So during fertilization, recognizing either it is the sperm of same species or different species, these glycoprotein and glycolipid going to work. If insulin receptor are these glycoprotein and glycolipid. Getting the things? So I told they act as a receptor, they have recognition of self and non-self, and these are always present toward outer side. And that is the reason we call cell membrane is a asymmetrical structure. Asymmetrical structure. Asymmetrical structure means you don't have symmetry. Why asymmetrical structure, if anybody going to ask? So you can say, sir, glycoprotein and glycolipid always present toward outside, never inside, and spectrine always toward inside. Getting the idea? Understanding the idea? So you got the structure of the plasma membrane or not? Tell me, dear children. It's completed? You got or get puzzled when I told more? Get puzzled when I told more? Got it? Is it clear? It's over? Can I move ahead? Yes. Now we are going to talk about transport across the plasma membrane. Transport across the plasma membrane. So if we talk about transport across the plasma membrane, have a look here. Plasma membrane involved in which one? Diffusion also. And plasma membrane also involved in active transport. Active transport. So diffusion occur toward the gradient. Toward the gradient means from higher concentration to lower concentration. No energy expenditure. No energy expenditure. No expenditure occur. So diffusion is of how many type? Two possible type through the plasma membrane. One is which one? Simple diffusion. Simple diffusion. Another is which one? Facilitated diffusion. Facilitated diffusion. Who undergoes in simple diffusion? Diffusion always from higher to lower concentration. Hydrophobic substances pass through membrane by which diffusion? Simple diffusion, and this simple diffusion occur through lipid bilayer. Simple diffusion occur through which one? Lipid bilayer or non-polar substances. If you talk about facilitated diffusion, it occur for which one? Hydrophilic substances and charged particle from high to low concentration. Polar substances from high to low concentration occur with the help of carrier protein. Occur with help of carrier protein. Which protein? Carrier protein. Carrier protein. So this is which kind of diffusion? Facilitated diffusion. And the third type of transport I told as active transport, it occurs against the gradient. Against the gradient. Getting the things or not? Against the gradient from low to high concentration. Low to high concentration. Okay, जी, energy expenditure occur. Energy expenditure occur, right? And you can say about pump protein participate. Pump protein participate. Plasma membrane also facilitate osmosis. Osmosis is what? A special kind of diffusion of solvent. A special kind of diffusion or kind of diffusion. You can say where who pass? Diffusion of water is called osmosis. You got the idea, isn't it? So these all kind of transport is facilitated by which one? Plasma membrane. So who is over, dear children? Plasma membrane is also over. Getting the things. Of course, example of active transport is what? Sodium, potassium pump. It is the example of transport of water is example of what? Osmosis. Sodium potassium pump is example of which one? Active transport. Now we are going to start about endomembranous system. And at least let me tell you about what? Introductory part of endomembranous system. So can we proceed? See, in long-term syllabus is too long. So you can't expect much. So we have to maintain the speed. And I told two things in a very clear manner. It is not mentioned in NCERT. Told na in detail about what? Desmotubule, plasmodesmata. Even I told about how cell plate is forming, who is forming it, how middle lamella is forming, who is forming it. That detailing I also give you. Okay, provided you. Okay, ji. Next. Endomembranous system. Endomembranous system. Actually, in this area, I just find like in plasmodesmata, students are simply struggling. They not have idea about how middle lamella is forming. Simply mug up. Middle lamella is cementing material between two cells. It is also called cell cement. What is the role of a teacher? You can mug up without me also, na? Getting the things. My role is where I did it. Got the idea how it is forming. Now I am going to talk about endomembranous system. Have a look here. Give your attention. So what is this endomembranous system? So what happened? See, each and every cell organelle are independent. Okay. But sometimes what's happened? Some of the cell organelle perform a coordinated function. So each and every cell organelle are independent. That is true. But what happened? But some of the cell organelle have coordinated function. Which function? Coordinated function. These cell organelles together called endomembranous system. Endomembranous system. These cell organelle are together called endomembranous system. It include endoplasmic reticulum, Golgi complex. It include endoplasmic reticulum, Golgi complex, lysosome, and vacuole. Okay. Cell organelles like mitochondria, plastate, peroxisome is not a component of endomembranous system. Component of what? Endomembranous system. Membranous system. Did you got it? Why? Because not have a coordinated function. Not have a coordinated function. So who is over? Endomembranous system over. In next class, I will say about the detailings of what? ER, Golgi body, lysosome, vacuoles. And later on, if time is there, another things, mitochondrial. Okay, Ji, it's over, it's done, clear? Any doubt? Especially in desmotubule, plasmodesmata, getting the things or not? Oh, yeah, sorry. Students are asking, tell about material. I forget to ask every day. Let me ask. Today, pakka. I am putting reminder for it. I just forget. Okay, bye-bye. Have a good day to all of you. I hope you understand the class. Clear? Over? Okay, Ji. Thank you very much.